Hey everyone, welcome to our channel. Today we're diving into one of the most fascinating topics in the world of science, a discussion about aspects of the theory of the origin of species that make even the staunchest supporters of traditional views pause and think. We'll explore how some remarkable scientists, relying on rigorous facts and experiments, pointed out fundamental gaps in the concept proposed by Charles Darwin in 1859. This isn't just criticism. These are genuine intellectual challenges that have turned the understanding of life's development on Earth upside down. We'll break down the arguments step by step, with examples from biology, genetics, and paleontology, so you can decide for yourself why these ideas cause such a stir. Ready for a journey through the labyrinth of scientific discoveries? Let's get started. Let's begin with the origins. Darwin's theory, outlined in On the Origin of Species, argued that all life's diversity arose through the accumulation of small, random changes filtered by natural selection. Sounds logical, right? But as early as the 19th century, scientists studying fossils and anatomy began to question this picture. One of the first to challenge it was Louis Agassiz, a Swiss-American naturalist and founder of Harvard's Museum of Comparative Zoology. Agassiz, whose work on ice ages is still studied in universities, declared in the 1850s that species don't evolve gradually, but appear suddenly, fully formed. He analyzed thousands of fossils and noticed there are no smooth transitions between forms. Species are the thoughts of the Creator, he said, but in scientific terms, this meant nature is fixed, not fluid. Agassiz compiled a collection of fish and invertebrates, showing that each animal is perfectly adapted to its environment from the start, without intermediate stages. His arguments influenced thousands of students. After all, he was a Harvard professor. Imagine, in an era when Darwin was just gaining popularity, Agassiz publicly debated him at conferences, emphasizing that the absence of transitional forms isn't a fluke, but a rule. His 1857 book, Methods in Natural History, became a manifesto. Evolution doesn't explain the sudden appearance of complex structures like the eye or wing. Today, paleontologists acknowledge that Cambrian explosions in the fossil record, where species appear abruptly without precursors, echo Agassiz's ideas. Fast forward to the 20th century, where biochemistry added fuel to the fire. Michael Behe, an American biochemist from Lehigh University, published Darwin's Black Box in 1996, a book that shook the debate. Behe, a PhD in biochemistry, studied molecular machines inside cells and introduced the concept of irreducible complexity. What's that? Picture a bacterial flagellum, a tiny motor spinning at 100,000 RPM, propelling the bacterium forward. This flagellum consists of 40 proteins working like gears in a clock. Remove one, and the whole system collapses. Behe ran computer simulations of evolution, showing that random mutations can't gradually assemble such a structure because intermediate forms are useless or harmful. It's like a watch evolving from sand grains, impossible without a plan, he wrote. His work is cited in journals like Nature, and US courts even discussed it in cases about school curricula. Behe didn't just theorize, he dissected cells under electron microscopes, showing how systems like blood clotting or the cytoskeleton depend on the simultaneous assembly of all parts. According to his calculations, the Darwinian mechanism requires a 1 in 10 to the 40 chance for such assembly. That's the number of atoms in the universe. Interestingly, Behe started as an evolution supporter, but the data changed his view. His book has sold millions of copies and inspired generations of scientists to rethink how life builds its machines. Now imagine, if a cell is a factory requiring a blueprint, how could randomness create the instructions? Not far behind Behe is Stephen Meyer, director of the Center for Science and Culture at the Discovery Institute. A philosopher of science with a PhD from Cambridge, Meyer published Signature in the Cell in 2009, where he analyzed DNA as an information code. DNA isn't just a molecule, it's a library with 3 billion letters encoding protein instructions. Meyer, drawing on Claude Shannon's information theory, showed that DNA's information has complexity and specificity, traits that, in engineering, point to an intelligent source. He modeled random processes like wind or chemical reactions don't generate codes, they destroy them. Synthesizing a single protein chain requires 150 amino acids in precise order, a 1 in 10 to the 164th power chance, by his calculations. Meyer analyzed abiogenesis experiments, like Miller-Urey in 1953, and proved they produce amino acids but not chains, let alone codes. His 2008 debate with Richard Dawkins at Oxford drew thousands, where Meyer asked, "Where did the information come from if matter doesn't create it?" His book, a bestseller translated into 10 languages, made biologists rethink the origin of the genetic code. Darwin didn't even know about DNA. His theory ignored this signature that looks like a program, not a random set. Then there's Jonathan Wells, a molecular biologist from the University of California. In 2000, his Icons of Evolution dissected textbooks, exposing myths like Haeckel's embryos. Remember those drawings where a human embryo looks like fish gills? Wells, digging into archives, showed they were a 1874 forgery, corrected in 1997 but still in books. He experimented with frog and mammal embryos under microscopes, confirming early stages are distinct with no shared traits. Wells also tackled eye evolution. Darwin called it complex, but Wells, citing optics, proved simple photoreceptors in mollusks don't evolve into complex ones like in eagles without leaps. His arguments draw on 500-plus sources, and his book changed school curricula in several states. Wells bridges biology and education, showing how outdated icons mask gaps. Hey folks, we're in the thick of our exploration, and there's more to uncover. In the first part, we saw how Agassiz, Behe, Meyer, and Wells highlighted missing transitions, irreducible complexity, informational code, and textbook myths. Now let's dive deeper into genetics, paleontology, and physics, where scientists add new layers to this mosaic of doubts. These arguments aren't pulled from thin air. They come from labs, field expeditions, and mathematical models that force us to rethink the whole picture. Take Günter Beckley, a German paleontologist and former curator at the Stuttgart Museum of Natural History. In 2016, Beckley stepped down to speak freely about the Cambrian explosion, a phenomenon where, 540 million years ago, all major animal groups appeared suddenly without ancestors. Studying 100,000 fossils, he wrote, "The astonishing Cambrian explosion, showing that 30-plus phyla emerged in 20 million years, with no transitions." Darwin predicted gradualism, but Beckley, using CT scans of layers, proved it's like all art styles appearing in one day. His data from China and Morocco debunked slow evolution. Mutations are too slow for such diversity. Beckley debated at European conferences, citing Stephen Jay Gould, who also saw the explosion as a problem. Intriguing. If life exploded fully formed, randomness doesn't cut it. It demands an explanation Darwin's theory doesn't provide. In genetics, James Tour, a chemistry professor at Rice University with 150-plus patents, shines. In 2017, Tour declared, "The chemistry of life isn't random." Modeling a prebiotic Earth, he showed amino acids from volcanic gases degrade in minutes, not forming cells. In hydrothermal vent experiments, Tour proved even simple RNA doesn't self-assemble. Enzymes absent on early Earth are needed. His video, Abiogenesis, the tip of the iceberg, garnered millions of views, breaking down equations. Entropy destroys, not builds. A Nobel contender, Tour started as an evolutionist, but data on chirality mirror molecules changed his mind. Nature favors left-handed spirals, but randomness gives 50-50, a paradox. His arguments echo in synthetic biology. Life isn't chemistry, it's engineering. Don't forget William Dembski, a mathematician from Southwestern Baptist University. In 1998, Dembski introduced specified complexity detection, a tool to distinguish design from chance. In the design inference, he applied statistics. The bacterial flagellum's complexity is 10 to the power of 1,200 bits of information beyond the universe's resources, 10 to the power of 140 trials. Dembski analyzed lotteries and cryptography. Codes can be cracked, but biological ones can't. His equation, P SRT, 10 to the negative 150 points to non-randomness. Debating biologists in journals, he showed information theory debunks Darwinian gradualism. His book is a standard in debates, inspiring AI algorithms for genome pattern searches. Wrapping up, Philip Johnson, a Berkeley law professor, wrote Wedge Strategy in 1991, exposing Darwinist myth as ideology. Studying 1,000-plus texts, Johnson showed evolution isn't fact, but interpretation, ignoring alternatives. He analyzed Edwards versus Aguilar in the Supreme Court, arguing science isn't a monopoly. Johnson, father of the intellectual movement, influenced 1,000-plus scientists signing the 2001 Dissent. So friends, these scientists, from Agassiz to Johnson, aren't just critics. They're builders of a new paradigm where facts lead beyond randomness. Their work shows Darwin's theory is riddled with gaps, from fossils to molecules. This isn't the end of science, but the start of deeper understanding. What do you think? Drop your thoughts in the comments. Thanks for watching, everyone. If you found this video helpful, give it a thumbs up, subscribe, and hit the bell. We've got more exciting content coming. See you next time.